Exciting news! You can now purchase my book, Rational Answers to Stupid Questions, simply by clicking the top link in the description. This will be your handbook for dealing with nonsense. Evolution, the Big Bang, creationism, flat Earth, it's all there in the book. The book is available exclusively on BookBaby, who has been my partner in creating my book. I've been wanting to write a book for years, but I had no clue how to get published. With BookBaby, you are the publisher. They will design your cover, give you professional editing, and are linked to all the major distributors. If you want to write a book, I highly recommend BookBaby. Enjoy the video.
But some people would tell you that going back to a common designer would leave common traits. Could be true, could not be true. I have no idea. But what I do know is that you're being disingenuous when you say that the only possible answer to two organisms sharing genetic traits is them coming from the same organism. That on its own.
I think I've repeatedly said the only logical inference you can make from the data is that. I think I also did say that's only possible if they share an ancestor. But you know, when it comes to things that are known so well, like that Earth goes around the sun rather than the other way around, when you say things like the sun sets, that's only possible if the Earth is round and is moving. I guess it could technically be true if the Earth were stationary and the sun went around it, but Earth would be round. Now, could it be that it's actually because of whatever bullshit those idiots make up? Sure, but nobody talks that way. Like nobody placates to every bullshit post hoc cope explanation.
So I'm just gonna ignore the flat Earth stuff because that is a whole different discussion that we could have some other time.
And you would lose.
But anyways, going back to evolution, what gives for example, when you look at the tissue in any organism, what gives the structure that the cells are placed in, for example? Like in connective tissue, for example. What gives the structure of your mesenchymal cells, or like the connective fibers that are
gene expression
in place?
The answer is gene expression.
So when a gene is expressed, what happens? A protein is made, correct?
Yeah. I mean, that's part of it, but oftentimes there's quite a lot of cell-to-cell communication, and there are environmental triggers that go into
and not necessarily. Yeah. Oh, my point is the actual placement of said proteins in specific tissues does not come from genes.
The what?
For example, when you're looking at white fibrous tissue.
Hold on. Say that again, because I missed it.
What I'm saying is, when a gene is expressed, the product protein does not get the information of where it is going to get placed in from the DNA.
No, it gets it from neighboring cells.
Yes and no. Because when you say neighboring cells, the very first cells that come by through embryology, where do they know where to go and where to allocate proteins?
Well, they don't do anything like that right away because they're either totipotent or pluripotent stem cells. I don't remember which term is the correct one. But you literally just divide until you're a morula, and then a blastula, and then a gastrula, or whatever.
It is
whatever. It's completely undifferentiated tissue.
Yes.
Only until you end up with an endoderm, mesoderm, ectoderm does the chemical signaling from like one cell to the next guide those cells in their differential development.
Which is exactly my point. The placement of specific tissues in specific locations comes through the mother having the baby inside of her.
Because
it doesn't come
because it implants on the uterine wall and I don't remember which layer it is. You start to form. It's actually interesting because the embryo is just a ball, but only the cells really like attached to the uterine wall develop into you. The rest forms the placenta and the amnion and a few other things. So what exactly is your point then? So because when the cell attaches to the uterus, that guides the differentiation of the cells. Therefore,
it guides the migration of the cells.
Yeah. Therefore, what?
Therefore, genes, for example, any mutation in a gene could result in the formation of a different protein. But that protein being used and utilized and then distributed along the body doesn't come from the DNA itself, the genetic code. It is reliant purely on the embryological development of the fetus in the mother. So when any evolutionary change that results in different characteristics comes by, like for example, this is purely, I know this would technically be a straw man, but just go along with the example.
So you're saying there are no genes that control for the embryo attaching to the mother?
The embryo attaching to the mother is not genetically based.
Really? That's a claim.
No. It is.
You got anything to back it up?
The fact that ectopic pregnancies occur, they don't necessarily attach to a specific point purely because of genetic sequence, markers. They can attach anywhere, depending on where the embryo ends up. So you can have a embryo attach well, in this sense, it would be a blastocyst attaching to the abdomen, in the peritoneum, in ovaries, in the fallopian tubes. It could attach anywhere. It doesn't necessarily have to be in the fundus of the uterus.
Here's a paper from the National Institutes of Health. In this regard, gene profiling studies performed in humans have identified numerous genes involved in the transcriptional regulation of uterine receptivity and embryo implantation. So there appear to be genes that help control for the embryo attaching to the inner uterine wall. Now you could argue, yeah, but they attach to other things too. But even if that's the case, there still appears to be within the embryo genetic controls to help it attach to something, right? So, I mean, this is initiated through genes in the embryo. But like, whatever, I still don't think this is a problem for evolution in any way.
I didn't.
But just because it's the mother's body that guides the differentiation of the cells into specific tissues. So what?
My point was that if any mutation occurs in a gene, the product protein is going to be different. Hence, for example, when I know, again, this is a straw man technically, but follow the thought itself. If a mutation causes some random person to have an extra arm inside the mother's body, the morphological change that would result in the formation of an extra arm would come through via the development in the womb itself. It wouldn't come by just because of the genetic mutation.
Yes, it absolutely would come about that. Just because it happens where it happens doesn't mean that the location is what caused it. That's insane.
So I have a question for you. If I were to immediately right now to replace all of your genetic code with that would result in you possessing the genotype that would possibly result in you having an extra arm, would you grow an extra arm?
Almost certainly not.
Exactly. Because it would necessitate the fact that it grows during castration the mother's
What did that prove?
That any morphological changes would result in anomalies in the process of development inside the mother's womb.
Yeah. Meaning that it's gene controlled, not uterus controlled.
No.
No.
For example.
How so?
For example. Urachal fistulas. Can you tell me what gene would result in that?
A urachal fistula?
I've never even heard that before.
I don't know.
It happens when the allantois
Fistulas
during
are holes in
embryology.
the body, right?
Yeah. Yes. During embryological development doesn't completely obliterate, resulting in a hole through the umbilical cavity that basically causes the baby to leak urine from its belly.
Okay.
What genetic marker causes that?
It's a failure of Hox genes to like appropriately develop the tissues or whatever. The most common birth defect is a cleft palate, and that's from, if you watch like an embryo's face develop, you start off as this ghastly looking thing, and then the tissue closes together around the front of the face, and it doesn't always close all the way, so you get that cleft palate. That's not the it's not the uterus causing that to happen.
It is not. That is not what I said.
We've known about Hox genes for a very long time.
That is not what I said.
Then what did
What
you
I was
say?
saying is morphological changes from the typical buildup of a human would come by through failed processes in the womb, not necessarily because of the uterus.
Sure.
So again, the
Therefore-
same way you wouldn't grow an extra arm just because you happen to gain the gene that might lead for you to gain an extra arm. A animal that gets a mutation or that passes along a mutation doesn't necessarily mean that mutation itself is going to cause any morphological changes, because the protein that would, like for example, again in white fibrous tissue, the collagen that is being used in that specific tissue, if that happened to change, it would still be placed upon the places where the white fibrous tissue is present, like in cartilages and etc. So the position of the protein being deposited isn't going to change, because the morphological processes that would result in this morphology would still be the same in the womb. So you can have changes in a protein structure that result in the formation of different primary sequences of proteins, but they do not produce any different morphological structures unless there's a problem in the embryological development in the womb itself.
So you're telling me that evolution is false because the only way an organism can change is for it to change as it's developing in the womb, but any of those changes will be harmful.
No, I'm not saying any of the changes are going to be.
I think that's exactly what you said. Paleo, what did I miss?
Nothing at all.
Yeah,
Sorry,
I'm pretty sure that's exactly what you said.
What I was saying is, again, I'm going to try to rephrase it.
Oh boy.
Sure, go for it.
So, any gene mutation
Go on.
Would result in the protein that is being produced to be deposited and being used in the same exact way as in the normal unchanged gene, because of the way that the body has developed in embryological development. But when morphological changes occur, that is purely of problems in said development. It doesn't occur just because of the gene that was introduced or changed.
Yeah, literally false. I don't know. I don't know what to tell you.
I don't even know what to say.
I don't know. Sciisms, if you want to come up, then I will gladly let you in to talk about this.
Come on, brother, get up here. Oh my god! I'm just like sitting here. I'm like, what? Like, bro, speaking a whole new language.
Yeah. Funny enough, in the Blind Watchmaker, I was reading parts of it yesterday. He talks about a lot of this kind of stuff. What this guy's talking about here sounds a lot like Evo-Devo, developmental evolution or whatever, which basically says, now mutations aren't the big thing. It's just changes that occur during embryological development, that's really what drives speciation. And natural selection wouldn't act very strongly on them because you're inside the mother being protected during that time. It's really interesting. I'm certain it's a combination of both things, but like we can see that changes in the gene expression in the embryo could lead to, like, for example, snakes have many more vertebrae than their close relatives do. Well, we know that gene duplications that cause like basically duplicated segments of the body occur. Like we know that happens. So that could have happened in a snake, and it's funny because a lot of people would consider that kind of like a macro mutation, right? Although it wouldn't change what it is, quote unquote. But that could happen in the womb, and so you get a long lizard, right? And then eventually they progressively start to lose their limbs and develop a weird kind of locomotion. We have very good transitional fossils of snakes showing us that. So there you go.
And also the changes within like their jaw as well.
Yeah.
Which is a defining characteristic of what makes a snake a true snake versus like a lizard.
Yeah, it's actually snake.
A snake. There you go.
Snake. I'm a snake. Sayism,
Are we
where
ready
are
for
you?
that?
He said he was switching devices, so
Okay.
let him come up here and explain. I know he knows a lot about this stuff, so I want to hear him talk about it.
Me too.
So, while we wait, have you
Go for it.
have you heard of Dr. What was her name? Dr. Jane Doe.
Dr. Jane Doe.
Jane Doe.
Holy shit,
Yeah,
dude.
Jane
There's no
Doe.
way.
Yeah.
There's no way, bro. Dr. Jane Doe. Holy
Hi, Ems. What a pleasure. You're having fun in a mood. So actually, the Jane Doe thing is interesting, and it has veracity as well. But yeah, Peterson, you were actually pretty good on Evo Devo being the sort of thing. The newest sort of addendum to the modern synthesis is Evo Devo, meaning that there is yet another hyphen, and the hyphen is basically saying that the environmental surroundings are about gene expression. So one could go as far, like if you listen to the podcast Big Biology or Night Science, they're having fun with the concept that genes don't do shit. They can only do things because DNA doesn't in itself do anything, right? It requires a cellular environment for it to actually be expressed and to cause the ending phenotype. The ending phenotype has actually wider variety than we ever thought it did. So there is a particular gene set can have great amounts of developmental plasticity and end up with a final different phenotype at different life stages, whether we're talking about embryological development or from tadpole to frog or adult form of any sort of organism. This is true across all the major kingdoms. So it is a growing area of enhancing the modern synthesis, which is basically, developed in the 40s, applying Mendelian genetics to Darwin's theory. Right?
This reminds me how they've found out that there are certain salamanders that they start off in the water with gills, but then when they become adults, they're fully terrestrial and they lose their gills. But there's something you can do where when they turn into adults, they retain their gills.
Right.
Isn't that called neoteny?
Well, neoteny would be the retention of youthful traits or like early life stage traits. So we think of neoteny with sort of modern mammals and humans as being retention of like puppiness sort of thing. But I think. So go ahead, make your point. Let's sort of go down the path.
What about the in the womb development kind of stuff? Like where did that go wrong? I mean, I've said what I can say. I don't know if there was something you remember where you're like, oh, well, actually, so and so and so.
I think one thing to bring up, which is becoming more and more of a field, is bioelectrics, meaning that uh, the actual fields around are what sets. So let's say, for example, tadpoles moving to frogs. If one surgically takes a tadpole and surgically moves the eyes around and the mouth around before it does the transition to adult frog, it still goes exactly to the normal frog shaped face. And it does so by sort of an equilibrium of bioelectric forces. Everything migrates around and keeps migrating until it hits this attractor state of the right orientation. And that's doing so independent of genetic instructions. There's no instructions say move 7 cells over, 4 cells up, that sort of pixel sort of thing. It's not that, nor is it chemotaxis, nor is it interactions with other cells. This is a bioelectric effect, and it's getting more and more sort of unwound as to what this means.
Cool.
And this is in the last 5 years that we've gotten into this extra part.